So we're going to continue with chapter 3. So you should be watching these videos after we did the class activity, either in person or virtually, about the cell and how the cell is like a factory. So now let's talk about the physiology of the cell, some key functions of our cells, including membrane transport, and then a little bit about protein synthesis. So the learning objectives here are to define selective permeability, diffusion, active and passive transport, to explain what happens to a cell when it's put in all kinds of solutions, to describe how substances move through a plasma membrane, and then to briefly talk about what we call the central dogma of biology, which is to talk about how information flows from DNA to RNA to proteins. So to start off, let's think about membrane transport. So as a reminder, solution is a solvent and solute, and our plasma membrane is what we call selectively permeable. So some things can get through easily and others can't, and it depends on their chemistry. So again, plasma membranes are made of phospholipids. The majority of that space is the hydrophobic part, the water-hating part. But they also have these little teeny head groups that are polar. Things that can move through the membrane are things that are small and nonpolar. So really easy, oxygen, all of it can get through, carbon dioxide, nitrogen gas, steroid hormones, these small nonpolar things, they have no problem getting through. Now, the issue becomes when we start to have things that are either polar, so things like water, some water can move, but not all of it, or ethanol, right? Some of it can move, but not all of it. That's what these arrows are showing you. Larger or uncharged things, really hard time for our large polar molecules, things like amino acids or glucose. And our ions are things that actually have a charge to them, impossible. So small nonpolar, good to go. If they're larger, they need help crossing the membrane, and that's where our proteins are going to come into play. So keep in mind that the principle of diffusion applies, which means that things are going to move from an area of high concentration to an area of low concentration until they reach equilibrium. So they're going to move, we say, down their concentration gradient. It's like when you're in a super crowded room and someone opens a door, you're going to leave the crowded room and go to the less crowded room until they're about equal in crowdedness. Okay? Molecules do the same thing. They're going to move from high concentration to low concentration until they're equal. Now at equilibrium, things are still moving. They're just moving at the same rate. And so for our cells, remember that salts can't move. Salts can't really move. The water, that's the thing that can kind of move, because salts have a charge. So if you put a cell in a hypertonic solution, in a very salty solution, what's going to happen is the water is going to want to move to where that salt is. The water is going to be like, oh, that's so salty. Let me try and help them out with more water. Let me water down the salt. And so the water is going to leave the cells and go to that super salty solution. Isotonic means that the solution on the outside is the same saltiness as the inside. And so in this case, the water going in is the same as the water going out. And hypotonic, this is when the saltiness is actually inside the cell and the water is going to come on in until the cell swells and maybe even bursts. So again, water is going to move to where that salt is super salty to equalize it. Let's talk about filtration. That's a passive process, and it's really important for A&P too when we talk about our kidneys, where it happens only across capillaries, where things are pushed by a pressure gradient, not a concentration gradient. Not super important for A&P1, but thought I would mention it for completion. Okay, so how do things move then if they are large or charged or polar? Well, they use transporters or channels. And these are our proteins that help move molecules across a plasma membrane. Transporters work by binding to that molecule, that solute, and then kind of carrying it across the membrane. It's going to involve a binding and a change of shape, so it's a slow process. Whereas channels are just like opening a door. Everyone rushes through as long as you meet the criteria of the channel. So, overview again, if things are moving down their concentration, if they're just lipid soluble, so small nonpolar things, they move by simple diffusion. They just go through the membrane, no problem. If, however, they're large or they're charged, they're going to use a carrier. And if it's moving with the concentration gradient, we still call it diffusion, but this time it's carrier-mediated facilitated diffusion. Carrier-mediated facilitated meaning it's using one of those transporters. If it uses a channel, we say the same thing, channel-mediated facilitated diffusion. If it's a channel for water, it gets a special name. This is what we call osmosis. So osmosis is when water moves through an aquaporin. Now, these processes can also work against a concentration gradient. So passive transport is when things move from high concentration to low concentration, doesn't need any energy, so it's a passive process, either simple, through a channel, through a transporter. However, if we wanted to move something against its concentration gradient, we want to take something from a low concentration and put it into a high concentration, we want to shove another person into that crowded room, that is active transport because it takes energy. And to do that, uses something called a pump. And it can be, most of the time, it's an ATP-driven pump. So most of the time, we're going to use ATP as our energy to push something against its concentration gradient, to pump it. But plants use light, and we can also use a coupled pump where one molecule moves down its concentration gradient while another molecule moves up. And the classic example of that is our sodium-potassium pump. So here we're going to use ATP to move both sodium and potassium against their concentration gradients. So we're going to put a bunch of sodium together and a bunch of potassium together, all using one molecule of ATP. Finally, we can also move things by vesicles. So we can move vesicles out of the cell with exocytosis or into the cell with endocytosis. And so exocytosis, you don't need to know the specifics, just know the direction. Exo, we're going to move things out of the cell. And for endo, again, you don't have to know the specifics or the different types. Just know endo, we're going to move things into the cell. Okay, so for that, we have finished our discussion of transport, and we'll do one last video about the central dogma.